Somewhere in the hours before the first light, in the warm interior of the stomach, a slow and steady kind of work is already underway. The cells lining the stomach wall, flattened and close-fitting, arranged in a continuous surface that the body has been maintaining since before it was fully grown, are being replaced, not all at once and not in any dramatic fashion, one by one, in a sequence too gradual to feel and too precise to mistake for anything other than deliberate. Specialized epithelial cells move into position along the mucosal surface, pressing gently into the places left open by the cells that came before them. The whole lining renews itself every 3 to 5 days, and the reason it has to is that the stomach's own acids are strong enough to begin dissolving tissue if the surface is left unwatched. The body does not leave the surface unwatched. Through the small hours, through sleep, through everything else that is happening at once, this quiet cellular replacement continues, the same process it has performed for every night of a life, without instruction, without acknowledgement, without needing any of the effort the day will later ask for. The acids in question are not a small matter. The hydrochloric acid produced by the stomach's parietal cells is concentrated enough to sit somewhere between pH 1, 5, and pH 3, 5, which places it well within the range that dissolves metal, given sufficient time and surface area. The stomach produces it in quantities large enough to break apart the proteins in a full meal, to denature and unravel complex molecular structures that would otherwise remain intact. Against the interior wall, this would be genuinely destructive if the interior wall were left undefended. What protects it is a layer of mucus, dense and slightly alkaline, secreted by goblet cells distributed throughout the stomach lining, a layer that interposes itself between the acid and the tissue, neutralizing what reaches it and slowing what it cannot fully neutralize. The mucus layer itself is gradually consumed by the environment it holds back, and so the cells that produce it are replaced. And so the cycle continues, calmly and completely, while the person whose stomach this is lies still in the dark. There is something particular about this kind of process, the fact that it requires nothing, no conscious direction, no effort of will, no memory of the previous cycle, or anticipation of the next. The stomach's lining does not know it is being maintained. The cells moving into place do not know they are completing a pattern. The whole arrangement simply proceeds, carrying forward a design that has been functionally the same for longer than the species has had a name for what it is. By the time the light returns and the first meal of the morning enters the stomach, the surface it encounters will be fresh. The work will already have been done. The stomach is not alone in this. Across the body, at every hour of the night, a similar kind of maintenance is occurring at scales and speeds that vary enormously, but share the same fundamental quality: ongoing, undemanding, self-sustaining. The nose, for instance, is doing something the body has been quietly perfecting for a long time. At rest, it is still cycling air, still passing the molecules carried in that air across a small and remarkably elaborate surface at the top of the nasal cavity, where millions of olfactory receptor neurons extend fine, hair-like cilia into a thin layer of mucus, waiting for contact. The nose at rest is still the nose in operation. It is reading the air it encounters, cataloging what arrives, assembling combinations of receptor signals that the brain has learned to interpret as specific and distinct smells. The number of distinct smells the human nose can distinguish is considerably larger than most estimates suggest. For most of the 20th century, the figure commonly cited was around 10,000, a round, plausible-sounding number that had entered the scientific literature without particularly strong evidence behind it. In 2014, researchers at Rockefeller University published a study in the journal Science that directly tested the boundaries of human olfactory discrimination. Rather than asking participants to identify named smells, the researchers constructed mixtures of odor compounds at carefully controlled ratios and presented participants with sets of 3 mixtures, asking them to identify which one smelled different. By extrapolating from the discrimination thresholds observed, the team calculated that the human nose can distinguish at least 1 trillion distinct odors. The actual number may be higher. The figure of 10,000, at that point, was set aside. What this means in practical terms is worth holding for a moment. 1 trillion is not 10,000 multiplied by a large number in a way that is easy to picture. It is 10,000 multiplied by 100 million, a ratio that does not map easily onto everyday scale. The olfactory system achieves this range not by having 1 trillion separate receptor types, but by working combinatorially. The human nose contains somewhere around 400 types of olfactory receptors. Each type responds to particular molecular features, the shape of a molecule, the distribution of its charge, the groups attached to its carbon backbone. When a complex smell enters the nasal cavity, many receptor types fire simultaneously in a pattern shaped by the exact molecular composition of what arrived. The brain reads the pattern, not the individual receptor. And because the patterns can be combined and recombined in an enormous number of ways, the space of distinguishable smells expands to a scale that the number of receptor types alone would not suggest. Walking past a bakery is for the olfactory system an exercise in parsing several layers of molecular information at once. The volatile compounds produced by the yeast during fermentation, the Maillard reaction products forming on the crust, the fatty acid derivatives released as butter heats, each one contributing to a pattern the brain resolves into something felt rather than analyzed. The label the mind applies is simple. The process behind it is not. The olfactory system is, by evolutionary standards, old. Detecting chemical signals in the environment is one of the more ancient forms of sensory processing available to animals with complex nervous systems, and the receptors the human nose uses belong to a family that has been present in vertebrate lineages for hundreds of millions of years. The particular 400 types the human genome encodes are the result of a long history of selection, retention, and gradual refinement, some inherited from ancestors who relied on smell far more heavily than humans do, some more recently emphasized, most of them still doing the work they were shaped to do. The nose in the dark room, cycling air, is participating in something very old. The body that contains all of this is also physically considerably stronger than it tends to appear from the outside. The skeleton, which bears the body's weight and protects its most vital structures, is built from a material that has been studied seriously by materials scientists for some time, not because bone is exotic, but because it is, by any reasonable measure of structural performance, exceptionally good at being what it is. Cortical bone, the dense, compact tissue that forms the outer shell of long bones like the femur, can withstand compressive forces of roughly 170 megapascals before it fractures. 1 megapascal is approximately 145 pounds of force per square inch. The number 170 held against that unit describes a material that is difficult to break by applying simple direct pressure. Reinforced concrete, concrete with steel bars running through it to improve its tensile strength, typically fractures somewhere between 20 and 80 megapascals under compression. Bone, adjusted for weight, outperforms it. The femur, the long bone running between the hip and the knee, bears most of the compressive load of standing and walking. In normal movement, it is subjected to forces that can reach several times the body's weight, higher when running, higher still when landing from a jump. The structure of cortical bone allows it to manage these forces not simply by being rigid, but by being slightly flexible. The collagen fibers running through the mineral matrix of bone give it a degree of elasticity that pure mineral structures lack. Under load, cortical bone deflects just slightly. The collagen bends. The mineral matrix distributes the stress across a wider area. Then, when the load is removed, the structure returns. This combination of stiffness and resilience, of resistance and slight give, is what makes bone so difficult to break under the kinds of forces normal movement generates. The geometry of the femur itself, slightly curved along its length, broader at the ends, narrower at the shaft, is part of what distributes load effectively. The shape is not incidental. It has been refined over deep time into something close to optimal for exactly the purpose it serves. All of this, the mucus layer, the olfactory receptors, the dense and slightly flexible femur, is held and supplied by a system whose scale is almost counterintuitively large for an object the size of a human body. The circulatory system, which moves blood through the body to deliver oxygen and nutrients to every cell and carry away what those cells produce as waste, is extensive in a way that the exterior of the body gives very little indication of. Blood moves through arteries, which carry it away from the heart. It moves through veins, which carry it back, and it moves through capillaries, the finest and most numerous of the vessel types, the ones that do the actual work of exchange at the cellular level. Capillaries are thin enough that red blood cells pass through them in single file, squeezing along channels sometimes barely wider than the cells themselves. They thread between individual muscle fibers, between cells and the walls of organs, into the tissue of structures as small and close-packed as the mucous lining of the stomach. Before arriving at the full scale of the circulatory system, it is worth spending a moment with the heart that drives it, or more precisely, with the specific cluster of cells responsible for keeping the heart beating. Tucked into the wall of the right atrium, near where the superior vena cava delivers deoxygenated blood back from the upper body, sits a small group of cells called the sinoatrial node. Individually, these cells are not remarkable to look at. Collectively, they do something that no other cell type in the body does in quite the same way. They generate electrical impulses spontaneously, without requiring any external signal to begin. The sinoatrial node is the heart's natural pacemaker, producing between 60 and 100 electrical impulses per minute in a resting adult, each one spreading outward through specialized conduction pathways across both atria, pausing briefly at the atrioventricular node, then moving down into the ventricles and triggering the coordinated muscular contraction that pushes blood forward. The signal generated by the sinoatrial node is not confined to the interior of the heart. It spreads through the surrounding tissue and into the larger electrical field of the body, producing a pattern of electrical activity measurable on the surface of the skin, with sensors placed on the chest, wrists, and legs. This is what an electrocardiogram records, not the heartbeat directly, but the electrical event that causes it, detected at the body's surface as a small rhythmic disturbance in an otherwise continuous field. The body broadcasts the signal whether or not anyone is listening for it. The sinoatrial node does not require external regulation to maintain its rhythm. It generates that rhythm on its own, with the autonomic nervous system adjusting the rate upward during exertion or stress and allowing it to slow during rest and sleep. At rest in a dark room, the rate drifts toward the lower end of its range. The signal continues. The chambers fill and empty. The blood moves. The sinoatrial node itself is, by the standards of what it accomplishes, very small. Its area in an adult human heart is roughly that of a grain of rice. The electrical impulses it generates are measured in millivolts, thousandths of a volt. The contractions those impulses initiate move somewhere around 5 liters of blood per minute at rest, a volume that rises considerably during vigorous exercise as the rate increases and the force of each contraction grows. Across a single day at rest, the heart moves something like 7,000 liters of blood through the body, a figure that is difficult to picture as a volume, but becomes slightly easier to hold as a measure of how much work the sinoatrial node, at its resting pace, quietly arranges. It does this every day. It has been doing it since before birth, when the fetal heart first began to beat. Driven by the same fundamental cellular mechanism, it will continue doing it without interruption until the body's long run is done. What the blood moves through as it travels from the heart to the body's extremities and back again is a network that continues to earn a kind of quiet astonishment when its extent is described in spatial terms. Every blood vessel in an adult human body, the large arteries leaving the heart, the medium vessels branching from them, the arterioles narrowing toward capillary width, the capillaries themselves in their enormous numbers, the venules collecting the returning blood, the veins carrying it back toward the chest, all of it, straightened out and placed end to end, would extend approximately 60,000 miles. The circumference of the Earth at the equator is approximately 24,900 miles. 60,000 miles of vessels is enough to circle the Earth 2.5 times, with a few thousand miles remaining. This number sits oddly against the body it belongs to. The body in question is roughly 5 to 6 feet tall, and its interior, however densely packed, occupies a volume that can be reasonably estimated and held in mind. The 60,000 miles is not a feature of the body's exterior dimensions. It is a feature of the density and extent of a network distributed through every organ, every layer of tissue, every centimeter of surface, a network fine enough that a single cubic centimeter of muscle tissue contains hundreds of capillaries, each one threading between individual muscle fibers to make exchange possible at the level where cells actually live. The vastness of the total length is not something visible from outside. It is folded inside the body the same way a great deal of what is most remarkable about the body is folded inside it, invisible, continuous, working, and as the capillaries in particular are worth lingering with for a moment, because it is the capillaries that account for the bulk of that 60,000-mile figure. Arteries and veins, for all their importance, are relatively few in number compared to the capillary beds they connect. A capillary is typically between 5 and 10 micrometers in diameter, a micrometer being 1/1,000,000th of a meter, a unit that requires some translation before it carries weight. A human hair, which is itself small enough that most people cannot see it on end, is roughly 70 micrometers across. A capillary, at its narrower end, is less than 1/10 the width of a single human hair. And yet these channels, too fine to see without magnification, too numerous to count individually, form the actual site of nearly everything the circulatory system exists to accomplish. Oxygen passes from the capillary wall into the surrounding tissue. Carbon dioxide and other waste products pass back in the other direction. Nutrients cross. Immune cells squeeze through in response to signals of damage or infection. Water moves. The exchange is happening right now in every capillary, in every organ, in the quiet of the early morning, as steadily as it ever has. The circulatory system does not distinguish between day and night in the way the body's other systems do. Digestion slows during deep sleep. Muscular activity drops to near zero in the most relaxed stages of rest. Even the brain, though active throughout sleep in its own particular ways, shifts its patterns of electrical activity significantly from waking to sleeping states. The heart rate slows, but the vessels remain open. The capillaries continue their exchange. The blood continues to move. Whatever the hour, the circulatory system is carrying oxygen to the neurons of the visual cortex, to the smooth muscle of the stomach wall, to the olfactory receptor neurons waiting in the nasal cavity for the morning's first complex smell. The femur is being supplied, its bone cells receiving the nutrients they need to continue the slow work of maintenance that bone undergoes throughout life. Old mineral removed, new mineral deposited, the structure quietly updated in response to the loads placed on it and removed from it across the day. All of this is happening in the body of the person lying still in the dark. The stomach lining slowly replaced, the olfactory receptors waiting at their posts, the femur sitting dense and reliable in the upper leg, the sinoatrial node sending its small precise signal through the chambers of the heart, the blood moving through 60,000 miles of vessels folded into a space no larger than a torso, a pair of arms and legs, a head. It is not happening because anyone is attending to it. It is not happening in response to anything the waking mind has arranged. It is happening because these are the processes the body has always run, because the design they embody has been present and functional for longer than the organism running it, has been recognizable as human, because somewhere in the layered instructions the body carries in every one of its cells is the entire specification for how to keep a human alive through the night, and the body follows it, reliably and quietly as it always has. There is a specific kind of reassurance in this that is different from the more general reassurance of knowing that the body is resilient. It is the reassurance of specificity, of knowing that the stomach lining is being replaced by identifiable cells performing an identifiable function. Compare that the olfactory system's range has been carefully measured and found to be larger than anyone expected, that the bone in the femur has been tested and found to outperform materials that humans deliberately engineer for strength, that the sinoatrial node's signal has been measured and recorded, and its mechanism described in enough detail to build devices that can replicate it. The reassurance is not that everything will be fine in some abstract sense. It is that everything is, in a very specific and documented sense, already being attended to, right now, in the dark, without any help required. The 60,000 miles is perhaps the most difficult of these facts to hold as an image, and also the one most worth trying to hold. An easier scale to begin with is the local one, the single cubic centimeter of muscle tissue, threaded with hundreds of capillaries, each one narrower than a human hair, the whole assembly too dense and fine to see without equipment that can resolve objects at the scale of micrometers. From there, the scale expands, the capillary beds of the stomach, the lungs, the liver, the kidneys, the brain, the skin, each organ's network vast in its own right, each one connected to the next through the vessels that carry blood from and back toward the heart. The total length of 60,000 miles is what these networks add up to when the body is understood, not as a structure glimpsed from outside, but as a network examined from within, from the perspective of the blood moving through it, turning through the smallest capillaries, pushing through the venules, climbing back through the veins toward the chest, arriving again at the right atrium, where the sinoatrial node is already preparing the next signal. This network, all 60,000 miles of it, is folded inside a body that can walk through a door. It sits, right now, arranged in the particular configuration of a person lying down, gravity pulling blood slightly differently than it does when the person is standing, the heart's workload reduced, the body's fluid distribution shifting in ways that sleep physiology has been charting for decades. The vessels are relaxed but open, the blood is moving, and it is warm, and it carries everything the body's cells need to continue their slow, continuous, unremarkable maintenance of the structures they collectively form. The stomach wall is renewing itself somewhere in this architecture. The olfactory receptors are cycling air. The femur is sitting at rest, its collagen and mineral matrix holding quietly, its bone cells working at whatever pace the night requires. And the sinoatrial node, smaller than a grain of rice, is sending its signal across the chambers of the heart in a wave that is, at this moment, too small and too interior to feel, but that has been doing exactly this since before the first memory, and that will continue long past the last thought of the night, out into whatever the morning eventually brings. 60,000 miles, folded inside one body, present and working in the dark, without drama, without announcement, without needing anything from the person it belongs to except the simple permission of lying still. And beneath a stand of Douglas firs in the Pacific Northwest, in the last gray hour before full dark, the soil does something that has no clear parallel in the world above it. There is no sound to it. There is no motion that any instrument placed on the surface could detect. But in the top few centimeters of earth, between the decomposing needles and the dense mineral layer below, a network of filaments, so fine they would be invisible to the eye, even held at close range, is carrying carbon from one tree to another, passing it through junctions and relay points along root surfaces in a slow, continuous transfer that has been going on for as long as the trees themselves have stood there. The soil is full of this. A single teaspoon of it, lifted and examined under sufficient magnification, would reveal several miles of pale thread folded into the space between soil particles and root hairs, threaded in every direction at once, branching and reconnecting and branching again in a structure too intricate to follow from any single point within it. What those threads belong to is not a plant, they are hyphae, the filamentous bodies of fungi, and in most forest ecosystems, they are essentially everywhere. They colonize the outer layers of tree roots, forming a sheath of fungal tissue around the root tip, and through this contact, they create a channel. Nutrients, water, and certain chemical compounds can pass along this channel in both directions, from tree to fungus, and from fungus to tree, or from tree to tree by way of the fungal intermediary. The fungi receive sugars from the trees. The trees receive phosphorus, nitrogen, and water that the fungal threads are able to gather from regions of soil the roots themselves cannot reach. Neither partner can fully thrive without the other, and in most temperate forests, the arrangement is so widespread that it is closer to the rule than the exception. For a long time, this was understood as a local and bilateral arrangement. One tree, one fungus, one root system. What the ecologist Suzanne Simard's research in the 1990s began to suggest was that the networks are considerably more interconnected than that. By tracing radioactive carbon isotopes through forest soil, Simard and her colleagues found that carbon fixed by one tree could be detected in the tissues of a neighboring tree within a matter of days, traveling through the fungal network between them. The implication, careful and still debated in some of its particulars, was that individual trees in a forest are not isolated in the way the surface view of a forest suggests. They are, at the root level, connected. The older and larger trees in a forest, sometimes called hub trees, or in the popular description Simard has used, mother trees appear to be nodes of particular density in the network, points where many fungal threads converge and where the volume of outgoing material is highest. What this means in practice is something like this. A Douglas fir seedling, germinating in the deep shade of its parent tree, should, by the logic of competition, be at a disadvantage. It cannot photosynthesize efficiently. It cannot gather enough carbon to build tissue quickly. It is too small to reach soil regions with adequate water. And yet seedlings in these conditions survive at higher rates than the light levels alone would predict. Simard's work suggests that one reason for this is that the larger trees are passing carbon into the network, not intentionally, in any sense the word usually carries, but structurally, because the network connects them and carbon moves along gradients within it. The seedling's root system, once it has formed mycorrhizal associations of its own, sits within a web that includes its parent, and the parent is, in effect, a source. The word for what the fungi form is mycorrhiza, from the Greek for fungus and root, and the relationship is ancient. Fossil evidence suggests that mycorrhizal associations existed at least 450 million years ago in plants that were among the first to establish on land. It is possible that the colonization of the terrestrial environment by plants was itself enabled by this arrangement, that fungi, already established in soil, extended the reach of early plant root systems far enough to make survival on land viable. If that is the case, then the forest floor in the Pacific Northwest is not merely old. It is a continuation of something that began at roughly the same time as the land itself began to hold plants, a relationship that has been refining itself for longer than there have been vertebrates anywhere on Earth. The network does not carry only nutrients. When a tree is under stress, drought, insect damage, fungal disease, it releases chemical compounds through its roots and into the soil. Some of these compounds travel through the mycorrhizal network to neighboring trees, where they can trigger changes in the receiving tree's chemistry. Whether this constitutes communication in any meaningful sense is a question that researchers have been careful around, because the word carries implications about intent and awareness that cannot be attributed to trees. What can be said more plainly is that the chemical signal travels, and that it arrives, and that the tree that receives it changes its behavior in response. It may increase production of defensive compounds. It may shift resources. The change is real, whatever the right word for its cause turns out to be. In the low light of a forest in the Pacific Northwest, all of this is happening at once, in every direction, in the top few centimeters of soil that a person would not bother to think about if they were passing through. The fir stands still, the hyphae move, not quickly, not in any way that compresses into drama, but steadily, in the old chemical language of gradients and diffusion, passing what they have to where it is needed. The whole arrangement holding together through a set of transactions so small and continuous that even the trees themselves, in whatever sense trees can be said to know anything, do not know they are in them. Some distance away, on a tropical reef, flat, at low water, an animal roughly the length of a human hand moves through a shallow crack in the rock and stops. Its eyes, mounted on stalks above the head, track an object a short distance away. What the eyes are doing in that moment is something that has only been understood relatively recently, and even now the understanding has the quality of an early map, detailed in some areas, blank in others, accurate enough to be trusted, but not yet complete. The mantis shrimp's visual system contains 16 types of photoreceptor cells. The human eye contains 3. The range of wavelengths these additional receptors can detect extends into ultraviolet and far into the infrared, covering parts of the spectrum that no vertebrate eye can access. For some years, this was described as giving the animal a richer visual experience than any other creature, a more complex, more colorful internal picture of the world. Later research complicated that. It turned out that the mantis shrimp's brain does not appear to integrate the signals from its 16 receptor types into a combined image in the way that the human brain does. Instead, it may read the receptor outputs more like a barcode, not blending the colors but categorizing them directly. A faster system, perhaps, than one that requires comparison and integration, but a different one. Whether the mantis shrimp sees more than a human, or simply differently, is not settled. What is settled is that it acts on visual information that is invisible to every other animal on the reef, including the prey it is stalking. The prey, in this case, is a small crab sitting in shallow water, and the mantis shrimp has apparently seen it and begun to move toward it in that way some predators have, very slowly, then not slowly at all. The strike, when it comes, is one of the fastest movements produced by any animal. The dactyl club, a dense calcified appendage folded beneath the head, accelerates to speeds of up to 50 miles per hour through a mechanism that does not depend on muscle speed alone. It works instead on the principle of a latch and a spring. Muscles contract slowly, storing energy in a curved saddle-shaped region of the club that acts as a spring, and a locking mechanism holds the club in place while energy builds. When the latch releases, the club snaps forward faster than any muscle could directly drive it, in a motion that takes roughly 1,000th of a second. The force produced by the strike is enough to shatter thick glass, but the strike is only part of what happens. As the club moves through water at that speed, the pressure behind it drops so sharply and so fast that tiny bubbles of water vapor form in the low pressure region, a phenomenon called cavitation. The bubbles collapse almost instantly, and when they do, they release a second pulse of energy, a shock wave, that arrives at the target a fraction of a second after the physical impact. The prey is struck twice in a single motion by 2 distinct forces. The crab registers the physical blow, then the cavitation shockwave, then nothing more. The mantis shrimp collects it and returns to the crack in the rock. The dactylclub itself has been studied by materials scientists with considerable interest. It is a composite structure, not simply a hard shell, but a layered arrangement of mineralized fibers, oriented in different directions, which allows it to absorb and redirect impact forces that would shatter a simpler material. The outer layer is dense and hard, suited to delivering force. Behind it, a helicoidal layer of fibers, arranged in a rotating pattern, absorbs the shock wave that would otherwise crack the club from the inside. The club strikes effectively thousands of times over the course of a mantis shrimp's life, without catastrophic failure. Engineers working on impact-resistant materials have found the architecture worth studying. Several research programs have drawn on its geometry in developing designs for body armor and helmet liners, materials that need to absorb sudden high force repeatedly without failing. The mantis shrimp itself is not thinking about any of this. It is sitting in its crack, processing what it sees through its 16 receptor types, waiting for the next crab. It has been doing this or something very close to this for approximately 400 million years. The order Stomatopoda, to which all mantis shrimps belong, is genuinely ancient. The body plan has remained recognizable across geological timescales that include several mass extinctions, ice ages, and the rise and disappearance of many of the animals that shared the reef with it along the way. The club in the spring mechanism and the 16 photoreceptors are not new inventions. They are a set of solutions to a set of problems that the ocean has been presenting for longer than there have been trees. By October, on the north central plains of North America, the air temperature has been falling for several weeks, and the milkweed has gone dry and brittle in the fields. Something is happening on a scale that is difficult to take in from any one location. Across a range that extends from southern Canada to the northern United States, hundreds of millions of monarch butterflies have begun to move in the same direction at the same time, south-southwest, angled toward a destination in the mountains of central Mexico that no individual butterfly in the current generation has ever visited, and that most of them will never have been within 1,000 miles of. They have never been to Mexico. Their parents did not go to Mexico. Their grandparents did not go to Mexico. In most cases, it is their great-grandparents who made the previous journey, who overwintered in the Oyamel forests in the mountains of Michoacan, and who returned north in spring. The butterflies in the October sky have received through their genetics a map to a place they have never seen. How the navigation works has been studied with considerable care over the past few decades. A monarch butterfly adjusts its flight direction relative to the sun's position, which is constantly changing as the day progresses. A fixed bearing relative to the sun in the morning would take a butterfly in entirely the wrong direction by the afternoon. What the butterfly does instead is account for the sun's movement across the sky by using an internal biological clock, a circadian rhythm that tracks time of day with enough precision to allow the sun's position to be used as a directional reference rather than simply as a light source. This time-compensated sun compass is located not in the brain, but in the antennae. Experiments in which the antennae were covered or surgically modified produced butterflies that could no longer maintain correct orientation, while butterflies with intact antennae continued to navigate accurately even in novel conditions. The endpoint of this navigation, the specific cluster of Oyamel fair forests in a mountain range in Michoacan, is not a large target. The overwintering sites cover a relatively small area at specific altitudes, and the butterflies that arrive there come from all directions, converging on the same hillsides in numbers that, in good years, coat the trees so completely that the branches bend under the weight. Researchers have confirmed that the navigational information must be encoded genetically because no individual butterfly ever makes the full round trip. The one that leaves Mexico in spring does not return in autumn. What returns is the fourth generation of that butterfly, carrying information it inherited through a chain of 4 reproductive events, a map passed forward, not through learning or memory, but through the structure of the genome itself. The journey covers up to 3,000 miles. Monarchs fly during the day, using the sun compass, and roost at night in trees along the route. They accumulate fat before departure and consume it along the way, stopping to nectar on late-blooming flowers when they can, but primarily burning reserves. The flight itself is assisted, where possible, by thermal updrafts and favorable winds. The butterflies drift with available air currents, using the sun compass to correct their bearing when the wind carries them off course. The whole system is remarkably adaptive. It fails when the climate shifts enough to disrupt the timing of the sun's position relative to the expected route, or when the overwintering forests are logged or degraded. But where conditions allow, The system functions with a precision that requires, from the butterfly's perspective, nothing more than following the instructions already installed in its antennae. Near the Oyamel Firs in late November, when the butterflies have arrived, the trees change color. From a distance, the firs appear to turn orange, the bark and needles obscured entirely by the massive wings. When the temperature drops at night, the butterflies cluster more tightly, and the rustle of wings in a resting colony is audible from some distance, a sound like slow rain. They are waiting, the way a great many things in the natural world are waiting, not for anything in particular, but for the temperature to rise enough to allow flight again, for the spring, for the signal from a clock they carry in their antenna, telling them it is time to move north. In a marine biology laboratory, somewhere on the Atlantic coast, a researcher is walking toward a row of large tanks. She has worked in this facility for several months. The octopuses in the tanks know this, or something that functions like knowing this, in whatever sense that distinction is useful. When she walks past the first tank, the octopus inside it does something it does not do for other staff. It moves to the front of the tank and orients toward her, its arms loosely arranged beneath it, its skin shifting through a brief pale pattern and settling. Some of the other staff it watches from a corner of the tank. Some it ignores. One person who sprayed it with water during an early handling session, an accident, not deliberate, but still a surprise, receives a response every time that involves the octopus sitting near the water's surface, apparently waiting. It has squirted water in that person's direction on several occasions since. The behavior has been observed in multiple laboratory settings and documented with enough consistency to have made it into the scientific literature, where it is described with appropriate care about what is and is not being claimed. Octopuses can distinguish individual human faces and respond differently to them. What makes this more than simply interesting is what else is known about cephalopod cognition. An octopus's nervous system contains approximately 500 million neurons, fewer than a human's, but comparable to the number in a dog's brain, and arranged in a way that has no parallel among vertebrates. Roughly 2/3 of those neurons are not in the brain at all. They are distributed throughout the 8 arms, clustered in ganglia at each arm segment, giving each arm a degree of local processing capacity that allows it to respond to its immediate environment without waiting for instruction from the central brain. An octopus arm that is severed from the body will continue to respond to stimuli for up to an hour, not because of residual reflex, but because the arm's own neural tissue can still receive and act on sensory information. The arms are not hands. They are something more and differently organized. 8 semi-autonomous problem solvers attached to a central coordinator, each capable of exploratory behavior, of threading into narrow spaces and returning useful information, of applying the approximately 2,000 suckers on a standard arm to grip, taste, and assess simultaneously. When an octopus investigates an unfamiliar object, the arms move through it independently, each one exploring a different surface, the information feeding back to the brain in parallel rather than in sequence. The result is a form of perception that is distributed in space in a way that the human equivalent simply is not. In laboratory settings, octopuses have been observed opening jars with screw-top lids, disassembling equipment they find aversive, navigating mazes they have learned, and solving multi-step problems in sequences that suggest retention of earlier steps. After a single brief training session, an octopus will recall the specific solution to a problem-solving task for several weeks. They are also deeply individual. Researchers who have worked with multiple octopuses over time consistently report that different animals have different behavioral dispositions. Some are exploratory, some are cautious, some are irritable, in a way that persists across sessions and cannot be explained by the specific conditions of any given day. Whether this constitutes something like personality in the sense the word carries for humans is not a question science can yet answer cleanly. What it can say is that the variation is real, that it is consistent within individuals, and that it affects experimental results enough that researchers have learned to account for it. The octopus in the tank watches the researcher walk past and then begins to move slowly through the water toward the back corner where it keeps the empty crab shells it has collected. It arranges one or two of them in a way that appears, to observers who have watched the footage carefully, to be purposeful. It has done this before. It is not clear why. The explanation that requires the least inference is that the shells are being used as a form of cover or den material. The explanation that requires the most inference is that the animal has a preference about how its space looks and is acting on that preference. The gap between those two explanations is not at the moment fully crossable. The octopus arranges its shells regardless. Somewhere in the Mediterranean, drifting at a depth of roughly 10 meters in water that is warm and clear in the way that October in that sea tends to be, something very small is doing something that has not been clearly documented in any other multicellular organism. It is smaller than a human fingernail, shaped loosely like a bell with trailing tentacles, and to a casual observer, it would look like any of several hundred jellyfish species in the same body of water. What it is doing is reversing the direction of its own development. Its cells already differentiated, already committed in the ordinary biological sense to the roles they play in a mature organism, are changing their identities. A muscle cell becomes something else. A nerve cell becomes something else. The organism is not dying and being replaced. It is converting what it already is back into what it was before. The process is called transdifferentiation. It is not the same as the way a stem cell becomes a specialized cell. That movement goes in one direction, from uncommitted to committed, from early to later. What transdifferentiation involves is a committed cell, one that has already reached its final form, reversing that commitment and becoming a different kind of committed cell, or returning to an earlier, undifferentiated state. In most organisms, this does not happen. The developmental pathways that convert stem cells into specific tissue types are generally considered to run in one direction only. The reason for this has to do with the layers of chemical regulation that surround the genome in a differentiated cell, marks on the DNA that indicate which genes are available and which are not, which settle into place as development proceeds and are not easily reversed. In the jellyfish Turritopsis dohrnii, something is removing or overwriting those marks, allowing the cell to read a different part of the genome, to return to an earlier identity, and to develop from there. The trigger for this reversal appears to be stress, starvation, physical damage, old age. When the adult medusa, the sexually mature form of the organism, encounters conditions that would ordinarily precede death, it begins instead to revert. The bell contracts. The tentacles are reabsorbed. The organism settles to a surface and attaches itself, beginning the transformation from mature jellyfish back to the colonial polyp form from which it originally emerged. From that polyp stage, it can develop again into a new medusa, genetically identical to the one that just reversed, born again from its own mature tissue. In theory, and the qualification matters, because this has not been observed through a complete cycle in controlled conditions often enough to treat it as established. The animal could repeat this process indefinitely. The life cycle does not necessarily terminate. Aging, in the sense of a progressive and irreversible movement toward death, does not appear to apply to this organism in the way it applies to most others. What happens instead is a loop: development, maturity, reversal, development again. The jellyfish is not immortal in the sense of being indestructible. It can be eaten, it can be damaged in ways it cannot recover from. It can fail to find food during the Medusa phase before reversal becomes an option. But the internal clock that most organisms are running, the one that counts toward an end, does not appear to be running in the same way inside Turritopsis dohrnii. Researchers studying the molecular mechanisms involved have found that certain genes associated with cell cycling and differentiation are active during the reversal in ways that are not fully understood. The process is not random. It proceeds through recognizable stages, producing recognizable intermediate forms, suggesting that it is regulated rather than chaotic. Something in the genome of this jellyfish carries instructions for how to run development backward, and those instructions are sufficiently stable to have persisted across evolutionary time without being lost. Other jellyfish species do not have them, or do not have them in a functional form. Whatever happened in the ancestry of Turritopsis dohrnii to establish this capacity remains, for now, an open question. The jellyfish itself is not, of course, aware of any of this. It drifts in the October water of the Mediterranean, its bell pulsing in the slow rhythmic contraction that all medusae use for movement, its tentacles extended for whatever might drift within reach. If the current carries it into colder water, or if the food supply thins enough, the reversal will begin, the bell will slow, the organism will settle, the cells will begin, the quiet process of changing what they are, returning through the gates of development they passed before, and starting over. There are several places in the Mediterranean where Turritopsis dohrnii has been collected and studied, and there are probably many more places where it drifts without anyone measuring it. Its populations are not, by most estimates, large. It is not a species that blankets the sea surface in the way some jellyfish do. It is small and translucent and not particularly easy to notice. It was first described scientifically in 1883, and its capacity for life cycle reversal was not confirmed until 1996, more than a century later. In the years since that confirmation, it has received considerable attention from researchers interested in cellular aging and the mechanisms that control differentiation, because whatever Turritopsis dohrnii is doing at the molecular level, it appears to be doing something that other multicellular organisms cannot. The Mediterranean holds many things that have been there for a long time without being particularly noticed. Its water is old in the way that enclosed seas tend to be, slower to exchange with the open ocean than an exposed coastline, warmer in summer, saltier than average, clear enough at depth that light penetrates further than in many waters. It has held its current shape for millions of years and has contained organisms recognizable as jellyfish for far longer than that. The jellyfish of the Mediterranean are not, in any meaningful sense, recent arrivals. They belong to a lineage so old that most of the groups of animals the word animal currently refers to had not yet evolved when the first medusae were drifting in ancient oceans. And somewhere in that water, right now, in the warm, clear column of it, Turritopsis dohrnii is drifting with its bell pulsing slowly, its cells going about what cells do, metabolizing, signaling, maintaining the internal conditions that keep the organism organized and coherent. Whether any individual one of them is moving toward a reversal, tonight is not a question that admits of a certain answer. What is certain is that the capacity is there, written into the genome, waiting in the chemistry of the cell, like a set of instructions that have not yet been called for. The forest floor in the Pacific Northwest is still doing what it was doing at the beginning. The hyphae are still threading between soil particles in every direction. The carbon is still moving. The older trees are still connected to the younger ones in a network that has no center and no edge, that began before the trees themselves were saplings, that will continue for some time after the trees are gone. On the reef flat, the mantis shrimp is in its crack in the rock, and its eyes are open, and its dactyl club is folded and latched and ready. Above the North American plains, in the dark, the monarchs are roosting in whatever trees are available along the route, their wings folded, their antennae still, their biological clocks running quietly in the dark, keeping time so that the morning will be navigable. In the Mediterranean, something very small is drifting where the current takes it, moving toward an ending it may not quite reach, folding back toward its beginning instead, the water around it dark and old and still. At the base of the Great Pyramid of Giza, pressed flat against the lowest course of limestone blocks, a surveyor running a careful hand along the foundation would find something that takes a moment to make sense of. The base of the pyramid stretches for 230 meters on each side. Across that entire length, the distance of 2.5 city blocks, the distance that would take a person 3 or 4 unhurried minutes to walk. The deviation in levelness from one end to the other is 2.1 centimeters, not 2.1 meters, not 2.1 feet, 2.1 centimeters, which is roughly the width of a thumb. The entire structure, built over a period of approximately 20 years, from around 2560 BCE, rises from a platform so precisely flattened and so carefully placed that even now, after 4,500 years of settling and seismic activity and the long pressure of its own extraordinary weight, the margin of error is smaller than the width of a human hand. There is a particular quality to that number when it is held for a moment, not recited, but actually held. 2.1 centimeters across 230 meters of stone. The people who built the Great Pyramid did not have steel instruments. They did not have digital levels or laser surveying equipment or any of the measurement technologies that would make such precision straightforward today. What they had was a deep and careful tradition of practical geometry, an intimate knowledge of how to read the position of stars and the behavior of standing water, and a workforce organized with a degree of coordination that modern scholars have spent decades trying to fully reconstruct. What they produced was a monument that remained the tallest man-made structure on Earth for 3,800 years. Not until the 14th century, when the spire of Lincoln Cathedral was completed in England, did any human construction surpass it in height. 3,800 years is a span of time so long that it contains within it nearly all of recorded Western history. The pyramid was already ancient when the Roman Empire rose and fell. It was already ancient when the first universities were established in Europe. It stood taller than anything else on Earth's surface. Through all of that, through every century of those 38 centuries, from the moment its capstone was set until a medieval English cathedral edged past it. That flatness at the base is worth returning to, because it is where the precision begins, and without it, nothing above would hold. The ancient Egyptians appear to have used water channels or trenches to establish a true horizontal plane across the building site, reading the surface of still water as a guide and cutting the bedrock down to that level with copper tools. The orientation of the sides is no less exact. The The four faces of the pyramid are aligned to the cardinal directions north, south, east, west, with an accuracy of less than 1/15th of a degree. 1/15th of a degree. The prevailing hypothesis among archaeologists is that this alignment was achieved by tracking the positions of stars, specifically circumpolar stars that never set below the horizon and whose positions relative to true north could be read with great care over many nights of observation. However it was done, the result is a structure that points toward true north with an accuracy that most compasses cannot match. The precision of it is quiet in a way that is hard to describe. It is not the precision of a machine which does not know what it is doing. It is the precision of many thousands of people over many years who understood exactly what the goal was and found over the course of all that work ways to achieve it that were more exact than they needed to be. The 2.1 centimeters was not required by structural necessity. The pyramid would have stood just as well with a base deviation of 10 centimeters or 20. The care is above and beyond the strictly necessary, which is one of the ways that care announces itself across very long distances of time. Roman concrete is a different kind of announcement, quieter in its way, and it is still being made. Along stretches of the Italian coastline and at harbor structures throughout the Mediterranean basin, ancient Roman maritime concrete that has spent the better part of 2,000 years submerged in seawater is, as far as researchers can determine, still getting stronger, not degrading, not holding steady, getting stronger. Modern Portland cement, the material used in virtually all contemporary construction, is damaged by prolonged exposure to seawater. The salt ions work their way into the material over time, and over decades or centuries, what begins as a robust structure becomes honeycombed with small fractures. The Romans used a different formula. They combined volcanic ash from a region near Naples called Pozzuoli with seawater and lime, creating a material that researchers from the University of California, Berkeley, and a team including scientists from institutions in Sweden and Italy, publishing in the journal American Mineralogist in 2017, found was undergoing a process that modern concrete cannot replicate. The seawater was not attacking the Roman concrete, it was completing it. As seawater percolated through the material over centuries, a mineral called tobermorite was growing inside the concrete structure, forming interlocking crystals within the matrix of the material and actually reinforcing it from within. The concrete was using the seawater as a raw material. It was incorporating what should have been its adversary and becoming, very slowly, more solid. Tobermorite crystals are platy in structure, growing in interlocking sheets that bind together the other components of the concrete around them. In modern Portland cement, the seawater equivalent of this process tends instead to produce materials that expand and crack the surrounding structure. The Roman formula appears to have produced a material in which the mineral growth caused by seawater exposure has the opposite effect, tightening rather than loosening, consolidating rather than cracking. A harbor wall built in the 1st or 2nd century CE, submerged in the Mediterranean for 18 or 1900 years, is in many cases structurally sounder than it was when it was new. The Romans did not know, in any technical sense, why their concrete worked as well as it did. The chemistry of tobermorite crystal growth was not available to them. What they knew was that pozzolanic ash combined with seawater and lime produced a material that held. They knew this from observing that their harbors stayed in place. They knew it empirically through long practice in the way that most knowledge was built before there was a framework for naming what the knowledge described. The formula was passed through generations of builders and engineers, refined through use, adjusted when it failed, and kept when it worked, until the material itself carried a kind of knowledge that no individual builder entirely understood. 2,000 years later, that knowledge is still legible in the structure of the concrete itself to anyone willing to cut a thin section and put it under a microscope. The Mediterranean is a useful place to think about long time because so many things are old there and because the oldness of them is not always obvious. Things that look like ruins are sometimes still working. Things that look intact are sometimes hollow. The distinction between the ancient and the present is less fixed at the edges than it appears from a distance. A harbor wall that has been underwater since the Roman Empire is not a relic. It is a structure that has continued to do what it was built to do, by slightly different means than intended, for longer than almost any other human construction on Earth. Into that same Mediterranean, in the 3rd and 2nd centuries BCE, Cleopatra VII had not yet been born. She would not be born until 69 BCE, which means that when she eventually came to rule Egypt, the Great Pyramid at Giza had already been standing for roughly 2,500 years. Not 250 years. 2,500. The pyramid to Cleopatra was an ancient monument. It was already old in the way that it is old now. Not recently built, not still new in any sense, but fully historical, already the object of study and wonder and religious significance. When she looked at it, she was looking at something as distant from her own time as the fall of Rome is from ours. This particular arrangement of dates does something interesting to the usual picture of ancient history, which tends to flatten everything before the Roman Empire into a single, undifferentiated antiquity. Cleopatra lived approximately 2,000 years before the present day. The Apollo 11 moon landing happened in 1969, which is roughly 2,030 years after the end of Cleopatra's reign. The pyramid was completed around 2560 BCE, which is approximately 2,500 years before Cleopatra was born, which means that she was, in terms of calendar distance, closer to the moon landing than to the construction of the pyramid she could have visited as a tourist. This is one of those facts that takes a moment to fully settle. Not because it is counterintuitive in retrospect, the math is simple enough, and the dates are well established, but because the mind's default organization of the past tends to group things by category rather than by chronology. Cleopatra and the pyramids feel like they belong to the same era in the way that the mind organizes the ancient world into a single epoch, but the ancient world was long. It contained within itself spans of time comparable to the entire length of history from Rome to the present. The people who built the pyramids were as remote from Cleopatra as Cleopatra is from us. That symmetry is exact, or near enough to exact, that the difference is negligible. She would have known the pyramids as ancient things, as structures from a civilization that was already, in her time, a subject of historical study. She was Ptolemaic, Greek in heritage, ruling a country with a civilization far older than her dynasty. She spoke Egyptian, according to ancient sources, which distinguished her from several of her predecessors on the Ptolemaic throne, and she may have spoken several other languages as well. She was a scholar and a diplomat, operating at the intersection of Greek, Roman, and Egyptian cultures at a moment when all three were in complex negotiation. The pyramids she could have visited were already as much a part of the background of civilized life as the Parthenon was to later centuries, present, enormous, old in a way that had passed beyond argument. What she saw when she looked at the pyramid, standing at its base, is not recorded in any document that survives, but the physical experience of it would not have been entirely different from the experience a person has standing there now. The same stone, the same dimensions, the same flatness of the base, the same alignment to the cardinal directions. What has changed is the casing, the outer layer of polished white Tura limestone that would have made the pyramid gleam in the Egyptian sun and that was largely stripped away in later centuries for use in other buildings. In Cleopatra's time, the casing may still have been largely intact. The pyramid would have had smoother sides, steeper and more uniform, catching the light differently, but the scale would have been the same. The 2.1 centimeters would have been the same. The care of the people who built it, readable in the flatness of the foundation, would have been the same. 500 years before Columbus reached the Caribbean, a group of Norse explorers built a settlement on the northeastern tip of Newfoundland, in what is now Canada, at a place the Norse sagas called Vinland, and that archaeologists have identified as Lance aux Meadows. The settlement consists of several structures, turf buildings constructed in a style consistent with Norse architecture of the late 10th or early 11th century, and an area where ironworking took place, identified by the presence of ironworking debris and a small forge. The iron found at the site was not produced from local sources in the way that indigenous North American peoples worked iron. It was smelted from bog ore in a furnace, a technology that did not exist in North America until European contact. The buildings and the iron were found by Norwegian archaeologist Helge Ingstad and his wife, archaeologist Anne Stein Ingstad, who excavated the site beginning in the 1960s after Helge Ingstad had spent years searching for evidence of the Vinland voyages described in the Norse sagas. The settlement at Lance aux Meadows dates to around 1000 CE, placing it approximately 500 years before the 1492 voyage of Columbus. The Norse presence in North America is now confirmed by the physical evidence, not merely reported in the sagas. The turf buildings are real, the forge is real, the ironworking debris is real. What remains less settled is how long the Norse occupation lasted, how extensive their exploration of the surrounding coastline was, and what happened to the settlement and its inhabitants. The sagas describe conflict with indigenous peoples, described in the Old Norse texts as Skraelings, and some accounts suggest that the settlement was eventually abandoned because the Norse could not maintain a presence against local resistance. But the details are not recoverable from the physical evidence at the site alone, and the sagas are literary documents as well as historical ones, shaped by the conventions of the genre and by the interests of the people who composed and preserved them. What the site confirms, plainly and without ambiguity, is that people sailed from northwestern Europe to North America around 1,000 years ago, built structures there, worked iron there, and left behind evidence of their presence that has survived in the turf of a Newfoundland headland for a millennium. The headland at Lance aux Meadows looks out over the Strait of Belle Isle, the narrow passage between Labrador and Newfoundland, where the waters are cold and the light in autumn has a particular low quality that anyone who has spent time on a northern coast would recognize. The Norse who built those structures would have spent time looking at that water. They would have been aware, in a way that their contemporaries in Europe were not, of the precise shape of a continental coastline that most of the world would not encounter for another 500 years. The image of the turf buildings low against that headland, smoke rising from the ironworking area, the Atlantic behind them, still holding the route home. It is an image that belongs to a specific kind of human experience, the experience of being somewhere new, somewhere far, with a known way back and an unknown extent in every other direction. That experience is old. It is not particular to any culture or century. The Norse at Lance aux Meadows were not the first people to know it, And they were not the last, but they are among the earliest people known to have carried it to that specific piece of ground, and the evidence of their having been there has lasted. There is a persistent assumption about life in the medieval period that tends to arrange itself around a few vivid ideas: that life was short, uniformly brutal, and entirely consumed by labor. The life expectancy figure that appears most often in casual discussions of medieval history, sometimes stated as 35 years, sometimes as low as 30, is almost always a figure for life expectancy at birth, which was dramatically reduced by high rates of childhood mortality. Adults who survived past the age of 10 or 12 could, in many parts of medieval Western Europe, expect to live into their 50s or 60s, sometimes beyond. The figure is real, but it is describing something specific, and what it is often taken to describe is something else entirely. The matter of how much medieval people worked is similarly more complicated than the usual picture suggests. Historians examining the church calendar of medieval Western Europe have found that the liturgical year was densely populated with feast days, saints' days, and periods of obligatory rest. These were not secular holidays in the modern sense. They were days on which agricultural work was not to be done, days structured around religious observance and communal gathering. The cumulative effect of this calendar, when applied to the actual number of days available for agricultural labor, produces an estimate that medieval peasants in Western Europe may have worked an average of around 150 to 180 days per year on agricultural tasks. This is substantially fewer days than the modern working year, which in many countries runs to 220 or 230 working days. The comparison is not a simple one, and not an argument that the medieval peasant's life was easy. The work, when it happened, was physically demanding in ways that most modern labor is not. The insecurities of harvest failure, of illness, of the particular vulnerabilities that came with being a laboring person in a hierarchical and often arbitrary social order were real and consequential. But the picture of a medieval life as one of unbroken toil from dawn to dusk, every day of the year, without rest or communal pleasure or any of the ordinary texture of a human life, that picture is not what the evidence describes. What the evidence describes, at least in part, is a life that had a particular relationship to rest. The feast day was not a vacation in the modern sense. It was a different kind of day, structured differently, organized around different obligations, religious observance, communal gathering, the particular rhythms of a village calendar. But it was a day when the fields were empty, and the work had stopped, and people moved through the hours at a pace that was not the pace of agricultural labor. The bells rang, the smell of wood smoke and food moved through doorways. People were together in the particular way that people in small communities are together when they are not working, which is a different kind of togetherness than the kind that happens when they are. The medieval village on a feast day in late autumn is an image that tends to resolve, when held for a moment, into something quieter than the usual image of the medieval world. The fields have been harvested and are resting under a low sky. The light in late autumn in northern France, or England, or the Rhine Valley, comes from a particular angle, flat and unhurried, the shadows long even at midday. The work is done for now. The year's agricultural cycle has passed its most demanding point and entered its long quiet. The village is not waiting for something to happen. It is in the middle of something that is already happening, the rest itself. The feast, the particular ease of a day that belongs to no one's schedule but the church's, in which the church has given entirely over to the absence of ordinary obligation. People who lived in those villages and attended those feast days would not have experienced them as unusual in the way that a modern person might experience an unexpected holiday. They were structured into the year, expected, prepared for in ways both practical and religious. They were part of the rhythm of the calendar in the same way that the rhythm of the agricultural seasons was part of the rhythm of the calendar. Rest was not a departure from the structure of life, it was one of the structure's expected and recurring forms. The continuity between that experience and other kinds of rest, the rest of an ordinary evening, the rest of a body that has finished its work, the rest of a mind that has followed something interesting for a while and arrived, without urgency at the end of it, is not difficult to feel. The capacity for rest, for the particular ease of a day or an hour in which the work has stopped and there is nothing immediately required, is not a modern invention, it is very old. The feast day, and the field at rest, and the smoke rising from the doorways of a November village in the year 1200, are a version of something that belongs to the whole span of human experience, visible at a particular moment, in a particular place, but not confined to it. The Great Pyramid was already 2,000 years old when the village was new. The Romans had laid their concrete harbors 1,000 years before that village was built, and those harbors were still holding. Rev Erikson had been dead for 2 centuries by then. The settlement at Lanzo Meadows long abandoned, the turf walls already beginning the slow process of returning to the ground that would preserve them for a future excavation. Cleopatra had been gone for 1,200 years, and the pyramid she had visited as an ancient monument was now 1,200 years more ancient still, sitting in the Egyptian sun in precisely the same location, still level at the base to within 2.1 centimeters, still pointing toward true north with an accuracy of less than 1/15 of a degree. The people in that November village did not know most of this. They knew the church calendar and the agricultural calendar and the particular geography of the land within a few days' walk of where they had been born. Their world was deep and detailed within its boundaries, as all worlds are. The feast day they were observing had names and associations, a particular saint, a particular story attached to the day, a particular quality of light in the church at that hour of the year. The bell that rang was a specific bell hung in a specific tower, with a specific tone that the people in that village had heard all their lives, and that they knew, without thinking about it, the way they knew the sound of the wind from a particular direction, what connects them to the people who built the pyramid, and to the Roman engineers who mixed pozzolanic ash into seawater, and to the Norse crew who built their turf structures on a Newfoundland headland and watched the Atlantic from the door, is not a single thing. It is not a philosophy, or a value, or a recognizable trait. It is something more ordinary and harder to name, the fact of having been people with particular skills and particular knowledge and particular habits of rest and work in specific places at specific times, doing specific things that have left some trace. The trace is uneven. The pyramid left the clearest trace of all. The feast day left almost none, except in the records of the church calendar and the estimates of historians working backward from what those records imply about the structure of time. But both are evidence of the same general thing, which is that the past was as full of actual people as the present is, and that those people were doing the ordinary and extraordinary work of their particular lives with as much care and attention as the work required. The pyramid stands. That is the fact at the center of all of it. 4,500 years after the surveyors pressed their instruments to the bedrock of the Giza Plateau and found the level line, after the quarrymen cut the limestone blocks and the workers set them in place one by one across 2 decades of effort, after Cleopatra stood at the base and looked up at something that was already older to her than Rome is to us, After the medieval feast days and the Norse ironworks and the Roman harbors slowly consolidating in their seawater, after all of that, the pyramid is still there, still flat at the base, still facing north, still taller than almost anything built before or after it, for longer than any other structure in human history. There is something in that persistence that is not threatening, and not triumphant, and not particularly instructive. It is simply what it is, a fact about an enormous thing that was made with great care and has lasted. The people who made it had lives that the pyramid outlasted by several thousand years in every direction. Their individual concerns, their particular days, the quality of the light on the Nile in the evenings of the years when they worked, these are not recoverable. What is recoverable is the 2.1 centimeters. What is recoverable is the less than 1/15th of a degree. What is recoverable is the structure itself, present and exact and patient in the desert, not asking anything of anyone who looks at it, simply remaining in the way that things made with that degree of care tend, over very long periods, to remain. The fields of the medieval village have been empty for 800 years. The feast day bells have been silent for longer than that. The Norse settlement at Lansof Meadows passed back into the earth quietly, its turf walls becoming turf again, waiting. The Roman harbor walls are still at the bottom of the Mediterranean, still in the slow process of becoming more themselves. The tobermorite crystals still growing in the dark water, unhurried, doing what seawater and volcanic ash and time have been doing to that material since the engineers who mixed it were alive. All of it is out there, in the dark and the earth and the water, persisting in its various ways. The pyramid in the desert, the concrete at the bottom of a warm sea, the turf outline of a Norse building under a Newfoundland field, the records of a church calendar that gave, for several centuries, a few hundred days a year back to the people who worked the land, all of it still present in some form, all of it evidence of something. And somewhere in a November village that no longer exists, the sound of a bell that no longer rings, the smell of wood smoke from a fire that has been cold for 800 years, the particular quality of a feast day afternoon when the work has stopped and there is nowhere to be, and nothing required, and the light is coming through the doorway at exactly the angle the season brings it, low and flat and unhurried, the same as it always has been, the same as it always will be, falling across a floor where someone sat at rest in the middle of an ordinary life and found, for a moment, that the day held nothing but itself. Somewhere in the space between two stars, there is an object 12 miles across that contains more mass than the sun. Not a collapsed remnant in any loose sense of that phrase, a precise, specific thing, a sphere of compressed matter, small enough to fit inside the boundaries of a mid-sized city, dense enough that a single teaspoon of its interior material would weigh roughly 10 billion tons on Earth's surface. Hold that size in mind against that weight. Let the numbers sit near each other without needing to resolve. The object is called a neutron star, and it exists fully in open interstellar space, spinning sometimes hundreds of times per second, radiating heat from a surface hotter than most stars at their cores. It is not a hypothesis. Several thousand of them have been catalogued. The universe produces them routinely in the aftermath of stellar collapse and leaves them where they form. The interior of a neutron star is one of the few places in the cosmos where ordinary physics gives way to conditions that have not yet been fully described. In 2022, physicists at Kent State University proposed, based on models of neutron star mergers and the way pressure waves propagate through ultra-dense matter, that deep within a neutron star's core, there may exist a state of matter in which quarks, the particles that form protons and neutrons, move freely rather than remaining bound. This state, called quark matter, would allow pressure waves to travel at roughly 0.58 times the speed of light. That would make the speed of sound inside a neutron star the fastest that sound in any material can theoretically travel. On Earth, sound moves through air at about 343 meters per second. Through steel, somewhat faster. Inside a neutron star, if the model holds, pressure waves move at nearly 200 million meters per second. The number is too large to feel in any ordinary way. What is perhaps more useful is simply the knowledge that somewhere in the night sky, objects exist whose interiors follow rules that required the construction of entirely new physics to approach. Neutron stars are also among the most geometrically perfect objects known to exist, because they form from the gravitational collapse of a rotating stellar core, and because any irregularity is smoothed by the enormous forces at work during that collapse. Some neutron stars are estimated to be spherical to within millimeters across a surface 12 miles wide. A sphere that size, if it were made of ordinary matter, would be visibly lumpy, irregular, shaped by geology and chance. A neutron star, shaped by gravity alone, can approach a smoothness that nothing in ordinary experience comes near. The surface is not rock. It is a crystalline lattice of atomic nuclei arranged under pressures that would compress any other material to something unrecognizable, and beneath it, deeper than anything on its surface could reach, the physics that governs it remains, in several important respects, an open question. There is a particular quality to the night sky when it is held as a set of distances rather than a flat field of points. The nearest star to the Sun, Proxima Centauri, sits roughly 2 light-years away, meaning the light arriving from it left that star 2 years ago before it crossed the gap and reached an eye on Earth. The Andromeda Galaxy, visible to the naked eye as a faint smear in certain dark sky conditions, is approximately 25 million light-years distant. The light arriving from it now left Andromeda when the genus Homo had not yet appeared on Earth, when the ancestors of modern humans were something else entirely, something that walked differently and had not yet carried fire from one place to another. That light traveled for the entirety of that interval and arrived, in this moment, at a particular retina or a particular telescope mirror, having crossed a gap that contains no useful comparison. The estimated number of stars in the observable universe lies somewhere between 200 billion trillion and 2 sextillion, a range so wide that naming either boundary does not settle the question, only marks the territory of its uncertainty. Carl Sagan, working to find a way into that number for ordinary comprehension, compared it to the grains of sand on all of Earth's beaches and deserts combined. The number of stars exceeds that count by a factor of somewhere between 10,000 and 100,000. Every grain of sand at every beach on every coast on Earth, and then all of the desert sand beyond, and then that number multiplied again and again until the multiplying itself becomes difficult to hold, and the result is something like the number of stars currently burning in the observable portion of the universe. Most of them have never been seen by any instrument. Most of them circle nothing, or circle things too small and cold to be called planets. Some of them are circled by things that, if they could be examined, might turn out to be very interesting. The fraction of them that have been studied in any detail is very small. The fraction that will ever be studied in any detail is at current rates of investigation smaller still. A beach at low tide has a particular feeling to it. Wet sand compressed underfoot, ridged by water, each grain small and distinct. Stand at the edge of any beach, anywhere on Earth, and then try to extend that surface across every coast and every desert the planet contains, and the number of grains becomes something the imagination can gesture toward without really reaching. Now extend that count 10,000 times. Now understand that each resulting unit is a star with its own mass, its own temperature, its own particular position in the structure of the cosmos. The breadth of what exists out there, in the most literal sense, is not something that compresses neatly into the few moments a person has to think about it before sleep. It rests more easily in the background, an open fact about the scale of things, present without requiring resolution. On September 5th, 1977, a spacecraft about the size of a small car left the surface of Earth and began moving outward. Voyager 1 was not the first probe launched into the outer solar system, but it became the farthest traveled. It passed Jupiter in 1979, Saturn in 1980, and continued beyond the reach of any direct observation, sending data home on a radio signal produced by a transmitter generating 23 watts of power, roughly the output of a standard refrigerator light bulb. In August 2012, after 35 years of travel, Voyager 1 crossed the heliopause, the boundary where the sun's outward streaming charged particles give way to the broader interstellar medium. The probe entered interstellar space, the first human-made object to do so, and kept transmitting. As of recent years, Voyager 1 is more than 24 billion kilometers from Earth. A signal sent from its transmitter, traveling at the speed of light, takes over 22 hours to arrive. Ground teams send a command, wait a day, and receive confirmation that it was received. The spacecraft that carries those conversations is a structure of aluminum and gold, built in the 1970s, still operational, still moving outward at roughly 61,000 kilometers per hour. It carries a gold anodized aluminum record attached to its exterior, containing sounds and images selected as a representation of Earth, greetings in 55 languages, music from multiple cultures, the sound of wind, rain, surf, and a mother's first words to her newborn child. The record was designed on the premise that something somewhere might find the probe and play it. The probability of that event is not calculated in any serious way. The record was made regardless. The probe carries it outward into interstellar space, past stars it will not reach for tens of thousands of years, the golden disk on its side turning slowly through a medium with an average temperature close to absolute zero. What is perhaps most ordinary about Voyager 1, and in that ordinariness most worth pausing over, is the size of the signal. 23 watts crossing 24 billion kilometers, arriving in the California desert as something almost unmeasurably faint, a whisper across a distance so large that even light takes nearly a day to cross it. The receiving dishes on Earth amplify that signal by factors that require scientific notation to express, and from the amplified result, engineers extract readings from instruments still functioning, still gathering data about the environment through which the probe moves. The solar wind is gone at that distance. The medium Voyager 1 passes through now is what fills the space between stars. Thin, cold, older than anything that formed within the solar system, largely undisturbed for billions of years. The probe moves through it and measures as it goes. Time does not pass at the same rate everywhere. This is not a metaphor about perception or about the way long afternoons feel different from short ones. It is a geometric fact about the structure of space-time, first worked out by Albert Einstein in the early part of the 20th century and confirmed experimentally in enough ways that it now forms the operating basis of several technologies that billions of people use daily without knowing it. The theory holds that the stronger the gravitational field, the slower time passes relative to a position where gravity is weaker. On the surface of the Earth, time passes slightly more slowly than it does high above the surface, where gravity's pull is fractionally reduced. GPS satellites orbit at altitudes of approximately 20,200 kilometers, where the gravitational field is weaker than at sea level. As a result, the atomic clocks aboard those satellites run fast, by approximately 45 microseconds per day, relative to clocks on the ground. 45 microseconds sounds like nothing. Over the course of a single day, accumulated positioning errors from that discrepancy would amount to roughly 10 kilometers of drift. Over a week, the GPS system would become unusable. The engineers who designed the system were aware of this. They programmed the satellite clocks to run slightly slow before launch, so that in orbit, running fast due to weaker gravity, they would tick at the same rate as clocks at the surface. The satellites are, in a real sense, pre-adjusted for the geometry of space-time before they leave the ground. Two identical clocks, one placed at sea level and one carried to the top of a mountain, both tick at what feels like the same steady rate. Neither one announces what is happening. The difference is real and has been measured directly, first by Hafele and Keting in 1971, who flew atomic clocks around the world on commercial aircraft and compared their readings to ground-based clocks afterward, finding the predicted discrepancy. The clock that traveled had aged differently than the clock that stayed, not by much, by an amount expressible in microseconds. But the amount it differed by was precisely the amount Einstein's equations said it would differ by, and the equations have continued to match every test applied to them since. What gravity does to the passage of time is not approximate, it is exact, in the way that geometry is exact, because that is what it is, a feature of the shape of space itself. The Moon is roughly 384,000 kilometers from Earth on average, its distance varying as it moves along an elliptical orbit. It is large enough to be visible in detail with a modest telescope, well mapped by now in its surface features, its craters and highland plains, and long frozen lava flows named and cataloged across decades of observation. For a long time, the Moon was understood to be dry, a place where the processes that produce water on Earth were absent, where the vacuum and the exposure to solar radiation would strip away any hydrogen before it could bond with oxygen into anything stable. Then, evidence began complicating that picture. In 2009, NASA's Ellscross mission directed a spent rocket stage to impact a crater near the Moon's south pole, Cabeus Crater, which sits in a region of permanent shadow. The crater's interior has not seen direct sunlight in billions of years. The impact sent a plume of material up into sunlight, and instruments analyzing the plume detected water ice. The confirmation was not ambiguous. Water ice existed in the shadowed crater, preserved by the absence of sunlight across a span of time longer than most of the geological history visible at Earth's surface. In 2020, a study using data from NASA's Sophia Observatory, a telescope mounted in an aircraft flying above most of Earth's atmosphere, detected water molecules not in shadow, but on sunlit portions of the lunar surface. That finding was more unexpected. Sunlit areas of the Moon are exposed to solar radiation, vacuum, and extremes of temperature that should preclude stable water. The presence of water molecules in those regions suggests that water on the Moon is more widespread and held in more varied conditions than the earlier model allowed. The picture is not yet complete. The mechanisms by which water survives on sunlit lunar terrain are still being examined. A permanently shadowed crater near the moon's south pole is one of the colder places in the solar system. No sunlight reaches its floor. The temperature there is estimated at around minus 170 degrees Celsius or lower. The ice that was found in Cabeus crater is old in ways that make the word old feel inadequate. Not thousands of years, not millions, but billions. The ice potentially dating to a period of the solar system's history, when comets and asteroids were delivering water-bearing material across the inner planets at a much higher rate than occurs now. That bombardment slowed. The water delivered to the moon in those early impacts migrated, over long timescales, to the coldest traps available, the permanently shadowed craters near the poles, where sunlight never reached and nothing disturbed the slow accumulation. The ice settled. The crater remained dark. The solar system continued its other business for billions of years, and the ice stayed. It is worth pausing on what it means for something to persist for that long without change. The ice in Cabeus crater does not know it has been waiting. It is not waiting in any sense that involves experience. It is simply old and cold and present, the same ice, or very close to it, that arrived when the solar system was young, held in a dark that has been continuous since before anything alive on Earth could have noticed it. The persistence is not purposeful. It is a result of conditions that happen to align: the crater's geometry, its polar location, the absence of atmosphere to carry heat inward from the lit surface. Those conditions held, and so the ice held too, resting in its dark at the edge of a world that has been circled by human eyes, and now by human instruments, for thousands of years without anyone knowing it was there. There is something in the scale of the cosmos that can feel, at certain hours, like too much to hold in a single sitting. The number of stars is too large. The distances are too wide. The density of a neutron star compresses too much into too small a space for ordinary experience to find a foothold. These are not criticisms of the cosmos. They are descriptions of what happens when a kind of thinking evolved for grasslands and social groups, and the distance between one water source and another is asked to hold numbers that have no natural upper limit. The thinking does its best. It uses the tools available: comparison, analogy, the image of a beach and all its grains, the image of a small car in interstellar space with a light bulb's worth of radio power, and something passes through the comparison that is not the full number, but is not nothing either. A sense of direction, a sense of scale, the feeling of having looked in the right direction, even if the full view is not available. Voyager 1 is out there now, in this moment, moving. The signal it is sending will take more than 22 hours to arrive at the receiving dish in California. The engineers who built it have, in many cases, retired or died. The probe was designed and assembled by people who are now grandparents or are gone, and it continues to transmit in the deep interstellar medium where no natural radio source produces anything at its frequency, and where the temperature is within a few degrees of the lowest temperature anything in the universe can reach. It moves at 61,000 kilometers per hour through a medium that offers essentially no resistance, and it will continue to move for tens of thousands of years after its instruments fall silent, carrying its golden record through the dark between stars, its dish still pointed home even when there is no signal left to send. The GPS clock correction happens every day, quietly, without announcement. The satellite rises above the horizon, the clock aboard it, adjusted before launch for a geometry that no one at sea level can feel, ticks at a rate fractionally slower than it would in the absence of that adjustment, slower by exactly enough to match the rate at which time passes on the ground. The positioning data it sends is accurate to within meters. Cars turn at the right intersections. Aircraft follow the right paths. Ships correct their headings. None of this requires the users of those systems to know that time is passing at a different rate, 60 kilometers above their heads. The geometry of space-time manages itself, and the engineers manage the geometry, and the correction is made every day in quiet arithmetic that keeps the numbers from drifting apart. The moon holds its ice in a dark that is older than the evidence of the earliest life on Earth. The crater's floor sits in shadow that has not been broken since long before the first single-celled organisms learned to use sunlight to split water molecules and release oxygen into an atmosphere that had previously contained almost none. The ice was there in its crater, cooling, before the trees that would eventually become coal deposits had grown. It was there before the dinosaurs. It was there before the fish that would become the ancestors of everything that currently breathes air on Earth had moved from water to land. It was simply there, cold and dark and still, undisturbed by any of the biology that was unfolding on the planet. It circled, preserved by conditions that the solar system arranged without intention and maintained without maintenance. When the instruments aboard the Else-Cross Plume analyzing spacecraft detected the spectral signature of water ice in the ejected material, the ice that had been waiting was perhaps 3 to 4 billion years old. The detection lasted seconds. The ice had waited for billions of years. Somewhere between the neutron star spinning in open space and the crater resting in its permanent shadow, the universe is doing a great deal that requires nothing from the observer. The stars burn, the probes transmit, the clocks aboard the satellites run at their adjusted rates. The ice in the crater stays cold. None of it is oriented toward any particular witness. It proceeds as it has proceeded, at the scales and speeds and temperatures that apply to it, with or without any instrument pointed in its direction, with or without any name given to it, with or without any mind that finds it restful or large or strange, or simply, on an evening like this one, worth considering before sleep. The pressure inside a neutron star would not be eased by any material that exists at ordinary scales. The matter inside it is so compressed that the electrons that normally define the outer boundary of an atom have been forced inward and merged with protons, producing neutrons packed against each other in a configuration that has no analogy in ordinary experience. The resulting object is stable, in the sense that gravity and the quantum mechanical resistance to further compression have reached a balance that holds across thousands of years. The star does not continue to collapse. It also does not expand. It simply persists, spinning, sometimes radiating, occasionally paired with another star in a binary orbit that will, over millions of years, bring them close enough together to merge, an event that produces gravitational waves detectable by instruments on Earth, ripples in space-time moving outward from the collision at the speed of light, arriving here as a vibration smaller than the width of a proton. The instruments that detected those waves, the LIGO detectors, one in Louisiana and one in Washington State, are precise enough to measure a change in the length of a 4-kilometer optical path by a fraction of the width of a proton. The measurement requires that the facilities be isolated from every local vibration, trucks on nearby roads, microseismic activity, ocean waves breaking hundreds of miles away. The isolation is accomplished with a combination of suspended mirrors, active damping systems, and the careful placement of the detector arms. Even with all of that, the signal requires careful statistical analysis to distinguish from noise. The first confirmed detection, in September 2015, matched almost exactly the waveform predicted by Einstein's equations for the merger of two black holes. The match was close enough that the mass, distance, and merger timeline of the event could be inferred from the signal alone. Two black holes that had been orbiting each other for billions of years, somewhere around 3 billion light years away, merged in a fraction of a second and sent a ripple through the fabric of space-time that crossed a billion light years of universe and arrived at a detector in rural Louisiana as a vibration lasting less than a second, the size of a fraction of an atomic nucleus. Someone noticed it. The instruments were calibrated to find exactly that. What the universe does quietly is perhaps the most accurate description of almost all of it. The stars burn, the neutron stars spin, Voyager 1 moves, the GPS clocks tick at their corrected rates. The ice in the lunar crater stays cold and still. The gravitational waves from ancient mergers cross the space between galaxies and arrive here almost imperceptibly at instruments built to receive them. The moon continues its orbit. The light from Andromeda continues its journey. The processes that have been running since the early moments of the universe's history continue to run, without pause, without maintenance, without any particular regard for the scale of time they are crossing or the distances they are covering. It is not indifference in any emotional sense. It is simply the way things proceed when they are very large and very old, and operating at scales for which the words large and old are already inadequate. Late in the night, with the sky very wide and the air still, the light reaching any upturned eye has been traveling for various intervals of time, some of it years, some of it thousands of years, some of it millions, depending on the source. The eye receives all of it at once, the near stars and the distant galaxies arriving in the same field without indicating the difference in their age. The light from a star 4 light years away arrives alongside light from a galaxy 2 million light years away, and both reach the eye in the same moment, the ancient and the recent side by side, indistinguishable by ordinary perception. The sky holds all of those distances together in a flatness that conceals what is actually a structure of enormous depth, every point at a different remove from the one standing beneath it. The permanence of the ice in the crater, the precision of the clock correction, the faintness of the Voyager signal arriving after more than 22 hours of travel, these are not separate facts that happen to share a category. They are different angles on the same large thing, a universe that is very old, very wide, and operating at its own pace, with or without observation, with or without anyone noting what it does. The noting, when it happens, is one of the more interesting things the universe has produced. The ability to measure the slowdown of time at altitude, or to detect a gravitational wave from a billion light years away, or to read the chemical signature of ancient water ice in a plume of excavated crater material. These are things the universe has come to, over time, by producing the conditions in which such measurement becomes possible. The instruments are part of the universe noting itself. The neutron stars and the lunar ice and the interstellar probe are all part of the same thing, varied in form, continuous in substance, old in ways that are, at this hour, simply restful to acknowledge. In a permanently shadowed crater near the moon's south pole, the ice that has been there since the early solar system is resting, as it has always rested, cold and dark and old and still, undisturbed in its crater, the shadow above it unbroken, the surface of the moon continuing its slow orbit, the earth below it, moving through another night, the light from other stars arriving from their long distances and continuing past without incident. The ice does not change, the dark does not lift. The waiting, if it could be called that, is very patient, the patience of something that has no experience of time passing, only the condition of remaining in the cold and the quiet, exactly as it is. Ruth Wakefield reached for the chocolate bar sometime in the winter of 1930, in a kitchen that already smelled of browned butter and warm sugar, and broke it into pieces over a bowl of pale dough. The sound of it, the clean snap of tempered chocolate, the rough, irregular edges left behind, was the sound of something ordinary, something that would not have registered as a decision at all. She was not experimenting. She was finishing a recipe, moving through the familiar sequence of a kitchen afternoon, and the pieces of chocolate she dropped into the dough were meant by her understanding of how baking worked to melt completely into the cookies and disappear. They did not disappear. What came out of the oven instead were cookies with soft, yielding pockets of chocolate that had held their rough shape through the heat, darker than the surrounding dough, slightly firmer, present in a way that melted chocolate would not have been. She served them at the Toll House Inn in Whitman, Massachusetts, where she and her husband Kenneth ran an establishment with a genuine reputation for good food, and the cookies were noticed immediately. Guests asked about them. The recipes spread, as recipes do, through conversation and correspondence, and the particular way that something very good travels between people who have tasted it. Within a few years, the recipe had made its way onto the wrapper of Nestlé's semi-sweet chocolate bar, where it has remained ever since, still recognizable, still more or less what Ruth Wakefield produced by accident on an afternoon she was simply trying to finish her work. She sold the recipe to Nestlé for a lifetime supply of chocolate. Whether that exchange was a fair one depends on how long she lived and how much chocolate she used, and the answer to that question is lost to the ordinary record-keeping of a small inn in Massachusetts. What remains is the recipe and the cookies and the fact that an entire category of baked good traces its origin to a moment of expectation not being met, to something failing to do what it was supposed to do and turning out to be better for it. The Toll House Inn no longer stands. It burned down in 1984 on the eve of New Year's Day, and the loss was noted in newspapers, partly because of what the building had once been. But the recipe survived, as recipes do, independent of the kitchen that produced it, independent of the woman who invented it, existing now in hundreds of small variations in home kitchens and bakeries and the back pages of cookbooks where someone's grandmother wrote adjustments in pencil decades ago. The accident became a tradition, and the tradition became so common that it no longer feels like anything unusual at all, which is, in its own way, a kind of completion. There is something in the chemistry of certain foods that resists the passage of time so thoroughly that the time might as well not have passed. In the tomb of a minor Egyptian official, sealed in darkness for 3,000 years, archaeologists have found honey that remains edible, not honey that was once edible and has since become something else, honey that is still honey, still amber, still faintly fragrant, still holding the chemical ratios that make it what it is. The jar was sealed in a chamber that was never designed to be opened, and the honey inside it waited in the dark for 3 millennia without changing in any essential way. The reason for this is structural. Honey's moisture content is too low for the microorganisms that would otherwise alter it to survive. Below roughly 17% water, the environment inside the jar becomes inhospitable to almost everything biological. Its sugar concentration by the same measure, is high enough to draw water out of any cell that tries to establish itself there through a process called osmosis, which is simply water moving across a membrane toward wherever the concentration of dissolved material is greater. The natural acidity of honey, its pH sits roughly between 3, 2, and 4. 5, depending on the floral source, adds another barrier. And the enzyme glucose oxidase, present in honey as a contribution from the bees themselves, produces trace amounts of hydrogen peroxide as a byproduct of its activity, which adds a slow, mild antimicrobial effect that persists as long as the honey remains sealed from too much moisture. These properties together are not a design in the sense that no one engineered them. They are the accumulated result of what bees do when they make honey, reducing nectar to a substance stable enough to last through seasons when flowers are not producing, through the deep cold of winter, through the long interruption between one harvest and the next. That the resulting material happens to outlast the civilization that collected it is not something the bees were aiming for. It is simply what the chemistry allows. The honey in the Egyptian tomb was still sweet. That fact is worth sitting with for a moment. The sweetness of something made in the fields of a civilization that no longer exists, still present, still tasting of what it always tasted of, in a jar that was closed before any living language had been spoken on the land where Rome would later stand. In large warehouses on the flat agricultural plains of Washington state, and in the cool river valleys of upstate New York, and in the orchard regions of southern England and northern France, apples are sleeping, not literally, but the conditions inside a controlled atmosphere storage facility are designed to hold the fruit in something very close to a biological pause. The temperature is kept just above freezing, usually somewhere between 1 and 4 degrees Celsius. The oxygen content of the air is reduced to between 1 and 2%, compared to the roughly 21% found in ordinary air. And the atmosphere is continuously circulated through scrubbing systems that remove the ripening hormone ethylene before it can build to the concentration that would signal the fruit to continue developing. Überschickholter Honoridungsserum U, and all apple harvested on a crisp morning in late September or early October will remain at approximately that same stage of ripeness 9 or 10 months later, in June or July of the following year, when it will be delivered to a grocery store and placed in a display that gives no indication of what it has been through. The apple in June was picked in October. The October it was picked in is still held around it in the form of the precise cellular state, the controlled atmosphere preserved. It is, in a sense, still that morning, still just arrived at the moment the farmer decided it was ready. There is nothing troubling about this. The apple is not diminished by the interval. The chemistry of ripening has simply been interrupted at a point where the fruit is good, and the interruption has been managed carefully enough that when the process resumes, as it does once the apple is returned to ordinary air and ordinary temperature, it continues from where it was, rather than rushing to catch up. The apple is not aware of any of this, which is perhaps the detail that makes it easiest to hold. It has simply been waiting in the cold and the dark and the thin air, and it will, in the end, ripen on someone's counter in the summer. And in October it will have carried with it through the whole long interval of winter. The people who run these facilities understand the chemistry of ripening in considerable detail, which is partly what makes the whole arrangement possible. Ethylene is produced by the fruit itself as it matures, and it acts as a signal both to the fruit that produces it and to the fruit around it, a chemical broadcast that moves through the air of an uncontrolled storage room and accelerates the whole chamber toward the same state. Removing it continuously is, in effect, a way of keeping the room from talking to itself, of preventing the conversation that would otherwise move everything in the same direction at once. The apples rest in the interrupted sentence, waiting for someone to let them finish. Coca-Cola arrived in the world in 1886 as a medicine. John Stith Pemberton was a pharmacist in Atlanta, Georgia, working in an era when the boundary between a pharmacy and an experimental laboratory was considerably less defined than it would later become, and the drink he produced and sold at the soda fountain of Jacob's Pharmacy in May of that year was labeled as a patent remedy for nervous complaints, headaches, and what the advertising language of the period called neurasthenia, a general category of exhaustion and dysregulation that served as a diagnostic home for a wide range of conditions the medicine of the time could not otherwise address. The original formula contained extracts from two principal sources, the coca leaf, from which cocaine is derived, and the cola nut, which contains caffeine. The name combined them both. Pemberton died 2 years after inventing it, in 1888, before he could see what the drink would become. By that point, he had already sold portions of the formula to various business partners in the particular piecemeal way that a man managing debt and illness sometimes makes decisions, and the consolidation of those partial interests into the company that would eventually become the Coca-Cola Company was accomplished not by Pemberton himself, but by Asa Griggs Candler, who acquired full control of the formula in 1888 and 1889 and proved to be the kind of businessman Pemberton had not been. The drink's transition from a medicinal tonic sold at a single soda fountain to a nationally distributed product was, at its mechanical level, a story about bottling contracts and distribution agreements and the infrastructure of delivery, about the practical question of how a liquid reaches a person who lives far from the fountain where it was invented. The coca leaf extracts were removed from the formula around 1903, when the legal and cultural landscape around cocaine had changed enough to make their presence in a popular consumer beverage a liability rather than a selling point. The formula has been adjusted at various points since, through changes in sweetener and formulation and the particular carbonation levels used in different markets. What it has remained through all of these adjustments is recognizable, the same general flavor profile, derived from the same general combination of ingredients, arriving in roughly the same form it has arrived in for more than 100 years. The original pharmacist's intent to produce something that eased the nervous system, something that offered a small interruption in the ordinary complaints of the day, has not entirely disappeared from the experience of drinking it, even now. The taxonomy of fruit is a subject that rewards patience and tends to produce mild disorientation in proportion to how carefully one follows its logic. A strawberry is not, in botanical terms, a berry. The part of a strawberry that is eaten develops from the thickened base of the flower, the structure botanists call the receptacle, rather than from the ovary of the plant, which is the tissue that technically produces a fruit in the botanical sense. The small seed-like objects embedded in the strawberry's surface, the ones that catch in teeth and give the fruit its characteristic slight roughness, are the actual fruits, each one a small dry acanthus, developed from one of the flower's many separate ovaries. The soft red body they stud is technically a false fruit, or an accessory fruit, or what some plant biologists call a pseudocarp. It has been eaten as a fruit by every person who has ever encountered one, and it will continue to be eaten as a fruit by every person who ever does, and the botanical classification changes none of that. It just changes the name. Bananas present the same kind of problem in the other direction. A banana is, by the strict criteria of botanical classification, a berry. It develops from a single flower with a single ovary. Its structure follows the three-layered pattern that defines a berry: the outer skin, which botanists call the exocarp, the soft edible flesh within it, which is the mesocarp, and the inner layer just around the seeds, which is the endocarp. That third layer, the endocarp, is thinner and softer in a banana than in many other berries, which is partly why the category feels counterintuitive when applied here. The seeds themselves are present in the cultivated banana, but they have been reduced through centuries of selective cultivation to the small brown specks in the center of the flesh that most people eat without noticing and would not, without prompting, identify as seeds at all. The wild ancestor of the banana contained seeds large enough to be noticeable and hard enough to require some effort to eat around. What has been cultivated toward over the long interval between the wild plant and the fruit that arrives in grocery stores today is a version with the seeds present but effectively removed from the experience of eating. They're but miniaturized, technically fulfilling their biological role in the plant's anatomy, while no longer playing any practical role in the experience of the person eating the fruit. The banana meets the definition. It always has. The definition was simply not one that most people thought to apply. The botanist examining a strawberry and a banana at the same time arrives at a picture that is, in its own quiet way, a useful one, the classification system doing exactly what it was built to do, following the structure of the thing rather than the common understanding of it, and producing results that are consistent and slightly beside the point of daily life, and completely correct. Language built for precision tends to do this. It reaches a truth that is not the truth most people were looking for, and leaves the thing named in a way that does not change it at all. It is worth returning for a moment to the way food carries time. The honey from the Egyptian tomb carries 3,000 years of it. The apple in the warehouse in June carries the October morning of its harvest, preserved in the cold and the thin air. The chocolate chip cookie carries a winter afternoon in 1930 and the sound of chocolate breaking over a bowl. Coca-Cola carries 1886 and a pharmacy counter in Atlanta and the particular optimism of a man who believed he had made something useful. Even the banana on a kitchen counter carries the long, slow time of cultivation, the centuries of someone selecting for this particular fruit, removing the seeds incrementally, generation by generation, until what remained was something smooth and sweet, and exactly this shape. Food is not the only place where time accumulates quietly inside ordinary things, but it may be the place where people encounter it most often without noticing. The meal on the table arrived from somewhere. The something it arrived from somewhere else. The chain of arrivals stretches back further than it is usually comfortable to follow, but the discomfort of following it is not the point. The point is something simpler and more restful than that: the sense that what is ordinary has been made, and has been made carefully by people and processes and arrangements that existed long before the moment of eating and will continue long after. Ruth Wakefield's accident is the clearest example of this, because the accident is documented, because it has a name and a date and a location, because the specific winter afternoon in Massachusetts has been preserved by the fact that what happened on it became something people wanted to tell. Most of the moments that produce the ordinary things around any kitchen are not documented in that way. The person who first cultivated the banana variety currently in wide use, the Cavendish, which replaced the earlier Gros Michel variety after a fungal disease eliminated most of the commercial crop in the mid-20th century, is not a figure whose name is remembered in the way Ruth Wakefield's is. The people who first discovered that honey did not spoil, who first sealed it in a jar and found it still edible when they returned, are not known at all. What they found out persists, as the honey itself persists, past the point where the people who found it can be named. There is something in this that seems, at this hour of the night, appropriate to rest with. The things that are close by and familiar, the food in the kitchen, the ordinary objects of a daily life, have been made by processes longer and more complex than the morning routines that use them suggest. The apple was held in October through the whole of winter. The honey was sealed in a jar when the world looked different in almost every way and has not changed since. The cookie was invented by someone who was simply trying to finish her afternoon's work and made something that outlasted the building she made it in. A kitchen counter in the early morning is by most measures an unremarkable place. The light comes in low, the surfaces are cool. Whatever is left out from the night before, a bowl, a glass, a bunch of fruit, is still and quiet in the particular way that familiar objects are still and quiet when no one is actively using them. A bunch of bananas on that counter is botanically a cluster of berries. The seeds inside them are present and too small to see. The three-layered structure of each fruit is intact. The exocarp is yellow, or just beginning to spot, or still faintly green at the stem end, depending on when they were bought and what the air in the kitchen has done to them. They are exactly what they have always been, meeting a definition they were never asked to meet, fulfilling a classification that has no particular relevance to their presence on the counter or to the morning that will, in a few hours, make use of them. The banana does not know it is a berry. This is not a remarkable observation, but it is a true one, and at this hour it has a certain quality that is worth noting without pressing too hard on. The fruit on the counter is not waiting for anything. It is not building toward anything. It is engaged in the slow, patient process of ripening, a process that, without intervention, will continue at its own pace through the whole of the remaining night, and through the morning, and through however much longer the fruit lasts on the counter before it is eaten or put away. The ethylene it produces moves quietly into the air around it. The sugars in its flesh continue their slow conversion. The peel continues its gradual shift from the green of the stem end toward the full yellow. That means the starch has finished becoming sugar, that the fruit has arrived at what it was working toward. None of this requires attention. None of it requires anything from the person who set the bananas on the counter before going to bed. The ripening continues regardless, as it always has, as it will again tomorrow morning when the kitchen is in use and no one is thinking about the botanical definition of a berry or the enzyme activity inside a jar of honey, or the October morning preserved inside an apple in a warehouse somewhere on a cold, flat plain. The world that surrounds the ordinary objects of a kitchen is very old and very patient and goes on doing what it does without waiting to be noticed. The honey was sweet 3,000 years ago and is sweet now. The recipe on the wrapper is still more or less what Ruth Wakefield made on an afternoon she was simply trying to finish. The apple on the counter in June was picked in autumn and has been waiting in the cold and the thin air for the morning that will use it.